So I'm really excited. It's my first time in Edinburgh, which is very exciting, and Scotland gives me a warm welcome, like every time I was here so far, rainy and misty, but still I'm really excited to be here. So what I want to talk today about is a topic that we have been working on in the last few years in my group at the University of Tübingen. Like Rick just mentioned, my background is mostly in ancient human genetics. So I've been working a lot on Neanderthals and archaic humans. We still continue this kind of work at the University of Tübingen, but we have also started with this new field of research that we call ancient pathogen genomics, where we basically take ancient skeletons from the past, from pandemics, epidemic outbreaks, thousands of year-old material. We extract DNA and reconstruct pathogen genomes from those to learn something about the evolution of pathogens and disease. So the story of most infectious disease-causing pathogens actually is quite a recent story in human evolution. So it doesn't go back to Neanderthals and millions of years of human evolution, but it mostly goes back to a time period that we call the Neolithic. So that was the time when humans settled down. So they were not hunter-gatherers anymore, but they started a sedentary lifestyle where they developed agriculture. And what they also did is they domesticated animals. And it is believed, at least today by many researchers, that from those animals that humans domesticated, they actually received a lot of the pathogens that cause infectious disease in humans today. And there's examples here: measles, smallpox, flu, tuberculosis potentially, plague, leprosy, pertussis, all examples where you would find the closest relative of this human disease in one of our domestic animals, and therefore it has been hypothesized that it was actually transferred from those animals to humans. And that's a process that is also called a zoonosis. And some people that have followed the media in the last couple of weeks, there has also been a very bad zoonosis that was happening in Western Africa. For example, this Ebola outbreak that just happened was also zoonosis from fruit bats to humans. So it's a very common practice for pathogens to enter human populations by actually entering from human hosts to from animal hosts to humans as a new host. So it's a very common theme. So then, as soon as those pathogens enter the human population, they might adapt to humans and might actually become a human pathogen. And then, of course, as soon as humans developed over time bigger settlements, large cities, civilizations, those pathogens found perfect breeding grounds to actually multiply and cause then what we call major outbreaks that we then call on a continental scale, for example, pandemics. And some of those pandemics, some of those large outbreaks, we also know from historical records, where it was actually so bad that actually lots of historians were actually writing about those outbreaks, and I've shown some examples here. For example, the plague of Athens in the 4th century before Christ. That was potentially an epidemic of measles. It's not really confirmed. We don't really know what caused it, but certainly it had a very strong impact on history. So the king of Athens, Pericles, at that time died. It was the time of the Peloponnese Wars, and actually the Persians won the war because of this outbreak in Athens. Another example is the Antonine Plague in the 2nd century after Christ in the Roman Empire. Again, we don't really know what caused this pandemic, so why millions of people died. We just know it from the historical records. It was a Roman doctor, Galen, who actually wrote a lot about it. He also actually got the disease himself and eventually died of it. And that, again, had a strong impact on the Roman Empire. It actually stopped the expansion of the Roman Empire because it got weakened because of so many people dying. And then there's 2 other pandemics where we basically know today what was the pathogen that caused those pandemics. That was Bubonic Plague, and I will talk about that a bit further later on. And that was the Justinian Plague, which happened in the 6th century after Christ. Again, it had a strong impact on the Roman Empire, especially the East Roman Empire. It actually stopped what we call the Antique and actually started the medieval time, the Dark Age. It's estimated that up to 50 million people died during this large pandemic. And then, of course, the very famous one that probably all of you are familiar with, the Black Death, which was this pandemic that happened in the 14th century. And again, it basically had a strong impact on medieval developments. It started the Renaissance. It actually killed up to 30 to 50%, people estimate, of all Europeans. So it was also a major pandemic. So this all sounds a bit like stories from the past. And of course, we know in the 20th century, people developed vaccination programs, people have antibiotics. So you would think that we should actually be ready to kind of deal with those infectious diseases. And in fact, in the 1960s, some doctors proposed that by the end of the 20th century, all infectious disease will be eradicated because of those powerful weapons. Of course, today we know that this was unfortunately not the case. We have, first of all, newly emerging infectious disease that were not known 30 or 40 years ago, that due to globalization are spreading in the world. Most famous example currently for sure in the media is Ebola, an infectious disease that we didn't know 30 years ago, but now is actually spreading in some parts of Africa. Other diseases such as HIV have even crossed from Africa into the Western world and caused millions of dead people every year. SARS, Hanta, Lyme disease, all examples of newly emerging infectious disease. So infectious disease is still a very hot topic within medical research. And the other problem we have is that some of those old pathogens that have actually plagued humankind probably for millennia, like tuberculosis, they have become antibiotic resistant. So some of those strains actually adapt to our weapons and become antibiotic resistant, so therefore making it very difficult to treat them. So they're actually re-emerging, they're coming back, so they're basically spreading in the world again. So besides this being a very still relevant and interesting field of research, and I think we have seen tremendous also new insights into kind of the diversity and the types of pathogens that we have in the world today, we know very little about the evolution of pathogens in the first place. So we know very little about how do they actually evolve to become a human pathogen, what happens early on during their adaptation, how fast do pathogens actually change over time, so what's their mutation rate, how fast can they actually adapt, for example. And we know also very little about those long-time host-pathogen interactions. So how does the pathogen adapt to the human as a new host, but also how do humans maybe adapt to the pathogen? So do we also change? Do pathogens actually change our own genome, our own genetics, that we actually also change over time as a response? And why do we know so little about that? That is mostly because if you want to study evolution, let's say human evolution, you would look at fossils. You would look at fossil humans and see how they have changed over time. But pathogens usually don't leave fossils. We don't really have fossil pathogens. If we have a fossil bacterium, it's a tiny little round cube. But then still you don't really know what it is. It's just a round ball, and you don't even know what species it would be. So you can't really say anything about how it has changed over time. And this is where we started with this new kind of topic or research field that we call ancient pathogen genomics. So where we basically go back in time, take ancient skeletons from the medieval time or earlier, take what is left of the genetic material from the pathogens out of those skeletons and reconstruct ancient pathogen genomes to give us some sort of basically molecular fossils. And using those molecular fossils, we can then estimate things like mutation rates. How fast do pathogens change over time? We can see how they have changed, how they might have adapted to the human host. And what we can also do, and that has been very important for historians, we can also provide the information what has actually caused those ancient pandemics. What were actually the pathogens that have been present in an outbreak, in a big pandemic hundreds of thousands of years ago, like, for example, the Black Death. And the Black Death was indeed our first study where we applied this new field and these new technologies to find out what in the first place has caused the Black Death, and then also how did the pathogen change after the Black Death over the last few hundred years. So the Black Death itself is this pandemic from the 14th century, which only lasted about 5 years, from 1347 to 1351. And it is estimated that it killed approximately 30 to 50 million people, so about 30 to 50 million Europeans during that time. The historical records indicate that the disease itself started somewhere in Eastern Europe, and it has been suggested that it started somewhere in the Crimean Peninsula, where there was a colony by Genoese people that was called Kaffa. And this colony was actually attacked by Mongols, how they called them at least. And they actually catapulted dead bodies into the city. And from those dead bodies, it seems that a pandemic, or this epidemic basically, spread. The Genoese had to kind of retreat, and during their retreat on ships, they carried this disease on those ships to Constantinople, to Messina, and later on to Genoa, as well as to Marseille. And from there, it spread potentially overland, maybe overseas to London, and also then other parts of Europe. So within 4 to 5 years, it has been basically all over the continent, even going up to Norway and Iceland. So for a long time, historians have debated what was the pathogen that caused this disease. And they have debated that because they had historical records that described medical conditions of people that died during that time, but that didn't really fit the pathogens that we had in the 20th century based on modern medical records. Most people had suggested it was Yersinia pestis. Yersinia pestis is the causative agent of bubonic plague. It's a gram-negative bacterium. However, other people have said that it could be Yersinia pestis. However, there is medical records from that 14th century that more sound like a filovirus, that could be like Ebola, for example, that causes hemorrhagic fever. Other people said that it might be anthrax. Other people said it was a virus which is extinct. We will never know what it was. So there has been a lot of debate what actually caused the disease. And that was also one of our interests, to look at ancient DNA to find out what was actual the pathogen that was present during that time. So what is bubonic plague? Bubonic plague is actually not a human disease. It's actually more a rodent disease. It's a pathogen that you find in wild rodent populations such as squirrels and marmots, also in commensal rodent populations such as rats and mice that live together with humans. And occasionally the disease spreads from wild rodent population to those commensal populations. And it jumps over, quite literally, by transmission by fleas. So what happens is the flea bites an infected individual. And the flea takes up those bacteria, Yersinia pestis, into its stomach. And inside the stomach of the flea, the bacteria produce a protein that actually blocks the stomach of the flea. So the flea can actually not swallow the blood. So what happens? The flea is starving, and it keeps on biting. And with every bite, it actually infects a new individual or the same individual again. So it actually spreads the disease. It can actually go over weeks and even months. Eventually, the rodent might die of this disease because they are basically infected and die from it. And then the fleas look for the new host. That would be the next warm body that would have 37 degrees. And if that warm body tends to be, or happens to be a human, those fleas actually jump on the human, and they might then bite the human and infect the human with the bacteria. The bacteria itself, they travel to the next lymph node. The lymph nodes get infected, they get big, and that's what's then called the bubo. And from those lymph nodes over the lymph system, the bacteria can actually spread all over the body. They infect all organs. They also cause necrosis. So digits, for example, kind of die, and eventually even hands and feet. They become black. That's also why it's called the Black Death. And then people that are infected, about 70% of them die within 7 to 10 days. So mortality is extremely high for this disease. So bubonic plague, however, is not the only form how people can get infected with this bacterium. There's another form that is called pneumonic plague. And this is when people actually infect other people. And that's not by biting them, but that's actually by droplet infection. So if you have an infected lung and you cough and somebody inhales the droplets with the bacteria, that person might also get an infected lung. And that's actually a very severe form, because when you develop the first symptoms, it's often even too late today to give antibiotic treatment, because mortality is really high, it's up to 90%, and usually people die within 1 or 2 days. So it's actually a pretty severe form of plague. So what do we know about the genetics of this pathogen? So this pathogen has 4 genetic components. So those are a big so-called chromosome, which is a large string of DNA, has about 5 megabases, or 5 million bases, and it has also those so-called plasmids, which are round DNA molecules that can be also exchanged between bacteria. In fact, 2 of those plasmids, Yersinia pestis acquired sometime recently in its evolution from some other bacteria. We're not really sure where they got it from, but the closest relatives of this bacteria, they don't have those 2 plasmids here. So they're specific to Yersinia pestis, and they are also important for this whole cycle of the flea and the biting and the rodents as well. If we then take DNA sequences, so whole genome sequences of Yersinia pestis from bacteria that we find in the world today, because we still find it in China, in Madagascar, even in the United States, we have Yersinia pestis today in rodent populations. And when we take those DNA sequences, we have about 150 that have been decoded so far, and built a phylogenetic tree, so basically a family tree of the disease. The family tree looks like that. You have 4 major branches that are human pathogenic strains, so strains that infect humans and have been shown to actually be infectious to humans. And we have one strain here, which is an outgroup called Yersinia pestis microtus, that has been shown to only affect rodents. It has actually been tested on people, so it didn't infect them, fortunately for them. And then there's basically an outgroup here, which is the closest non-Yersinia pestis relative, which is a soil bacterium that's called Yersinia pseudotuberculosis. So what people have then also done in the past is they have tried to use this family tree to calculate when those different branches, when those different strains evolved. So when did they actually emerge in the first place? So when did, for example, the human pathogenic strains diverge from the rodent pathogenic strains? And they come up with pretty big numbers. So 700 years maybe ago, maybe 8,000 years ago, 2,000 years ago, maybe 30,000 years ago. So it seems not so easy to use this modern data to calculate those divergence times. And that is because if you do this, what you usually need are fossil calibrations, and that's not what the people have, as I mentioned before. So what we wanted to do is we wanted to enrich and enlarge this data set of modern Yersinia pestis with ancient Yersinia pestis from the Black Death to actually see how do they relate to those modern genomes and how do they fit into the family tree of modern Yersinia pestis to learn something about its evolution and how it might have changed over time. And the material to actually get those ancient pathogens comes from London, from a site that is called East Smithfield. So I guess most of you have been in the capital of England? No, UK, I guess. Sorry. And so this is the Tower of London here. This is the city map of London. And you have this site here, which is very close to the Tower of London, which is called East Smithfield. At least in the 16th century, it was called like that. Today, it's part of the financial district. I believe one of those cucumber buildings is on top of that. And when people were building those new skyscrapers in the 1980s, they actually recovered a cemetery close to the Tower of London here that they knew from this historical records was this site called East Smithfield. And from the historical records, they also knew that this cemetery was used only during the Black Death, because in the city of London, they had so many dead people that they had to dispose them somewhere. And they basically bought fields around the city to dump the dead bodies. And we had access to this collection, and we took 50 samples from the cemetery, where we now believe that most of the people died during the Black Death. And in 20 of those, we could at least show that Yersinia pestis DNA is present. So we were pretty certain that the DNA that we get out of there was from those Yersinia pestis bacteria. However, we don't just wanted to say in a qualitative approach, yes, Yersinia pestis was present during that time in London to show that, yes, it was the disease that caused the Black Death. But we wanted to go one step further and also wanted to reconstruct the genome. However, to do that, we have a major challenge. And that challenge is if you take DNA from such an ancient skeleton, and usually we use teeth because teeth are basically like a little box or capsula that has kind of protected DNA from degradation over time. So what you get from inside the tooth is still a little bit of like dried nerve tissue, maybe a bit of dried blood that you can extract from those medieval teeth. But the DNA that you get out of such teeth is not just pathogen DNA. In fact, most of that DNA that you get out of an ancient skeleton is not even human DNA, but most of the genetic material that you get from ancient skeletons are bacteria, fungi, plants, so all kind of microorganisms that were living in the soil, that were living in the skeleton, that were living in the site in London over the last 600 years. So it is quite challenging to actually get the few molecules out of this complex soup of DNA that belong to the pathogen. And to do that, we developed an approach that we call molecular fishing. And this molecular fishing is very much like if you go fishing here on a lake. So you basically have a pond where you have your fish, and you want to catch those fish, so you need a bait. And the bait that we use is what we call a microarray. So a little piece of glass that has on its surface DNA molecules attached. And the wonderful thing about DNA is that DNA has those double strands. And those double strands, they attract each other if they are complementary. So if there's an A facing a T, if there's a G facing a C. So if you have the right sequences that match each other, they attract each other and they pull each other. So what we designed on this piece of glass was the entire genome of modern Yersinia pestis. And we incubated then this piece of glass with the DNA from those skeletons. The fragments that are now complementary, that have a similarity of up to 90% to the DNA on this piece of glass, will bind to this piece of glass, and then the rest can be washed away. So by that, we fish out the fragments that we are interested in that look like Yersinia pestis. And afterwards, you can basically take this DNA off, you put it into one of those sequencing machines, and those sequencing machines are actually what make all this work that we do possible. Those are the newest generation of DNA sequencers that produce billions and billions of DNA sequences for very little money. And it's actually amazing. It's only 10 years ago that we used machines where the first human genome took 13 years to be sequenced, and it cost 2 billion euros to sequence it, whereas those machines can now sequence a human genome in just one day for 500 euros. So basically from 2 billion euros, we went down to 500 euros because those machines have improved so much. So they are really basically the reason why we can do all this kind of work, reconstruct Neanderthal genomes or also reconstruct those ancient pathogen genomes. If you then look at how much pestis DNA you had before this fishing approach, you see we had very, very little pestis DNA, whereas after this enrichment, we have a lot of pestis DNA. So we have fished out all those fragments. And those fragments we can now use to assemble the genome and reconstruct the genome of those ancient plague bacteria. However, before that, we have to be concerned about something, and that is very, very important in our type of research. And that is the authenticity of that DNA. Can we actually show that this DNA is really ancient DNA and is not some DNA which could be from the soil, from the lab, contamination? Maybe some people have just touched the bone and left some DNA in the bones that we have actually looked at. And to test the authenticity of ancient DNA, we look at something that we call DNA damage. Because DNA, like everything else in nature, degrades over time. So the DNA basically breaks apart due to chemical reactions that change the DNA over time. And when we look at ancient DNA, those tiny little molecules that we still have in those ancient skeletons, we see that especially at the ends of that DNA molecules, you have a lot of those DNA damages. Mostly you have damage where C's in the DNA become T's. And that is something that has been observed already 15 years ago. However, what we did not know 15 years ago is that those damage molecules leave a very specific profile. They leave a profile we call sometimes the smiley, even though that looks more like a grin than a smiley. But it's basically this pattern where you see, if you move along the DNA molecule, that at the beginning of the DNA molecule, you have a lot of DNA damage, and at the end, you have a lot of DNA damage. And that's something that we have not only observed in this Neanderthal DNA here, we have seen it in mammoth, we have seen it in cave bear, we've seen it in ancient pathogens, we've seen it in all kind of ancient DNA sources. However, where we do not see it is in contamination. So if DNA is from an archaeologist, is from a person in the lab, if it's DNA that is 5 years old, 50 years old, that DNA does not have damage. It's just not old enough, so it hasn't damaged yet, or hasn't been damaged. What we also did is we looked at this amount of damage, how it accumulates over time. So this is a series of samples. It's mostly cow bones, some of it are also horse bones, some of it are also monkeys from museums. And they span from 20 years to 100 years to 500 years to 10,000 years to 50,000-year-old material. And here on the y-axis you have the amount of damage. And you can see that there's a very good correlation. So the older the material gets, the more damage it accumulates. And you can also see that if you have material that has more than 20% damage, it is usually older than 500 years. But it is never younger than 100 years. So if we see 20% damage or more, we can be very certain that that DNA is old DNA. That must be DNA that is older than 100 years and should be then the real DNA from the skeleton, for example. And indeed, if we look at the human DNA from London, from the East Smithfield site, we first look at the human DNA, we see that it has this typical smiley shape, so the human DNA is old. We see it has about 22% damage. If we go back here, 20% damage, we're somewhere in the 500-year range. The samples are 600 years old, so that matches pretty well. If we look at the pestis DNA in the same sample, we see the same, we see the smiley here, so we can actually show that this pestis DNA, so this ancient bacterial DNA must be old. It must be from the medieval time. It cannot be from the lab contamination, for example, from the last 100 or 50 years or so. So now that we can say that there is authentic ancient DNA, we can use this ancient DNA to reconstruct the genome of those ancient bacteria. And we were able to do that. We could reconstruct about 99% of the ancient East Smithfield Black Death bacterial genome. We have seen every position in that DNA about 30 times, so we have a pretty good quality DNA sequence. And now that we have the entire genome, we can compare this ancient genome to the modern ones, to put it basically into the family tree that I've shown you before. So now you can look at this family tree again, and now you can think, where might the ancient genome fall on that tree? So I don't know if you have some hypothesis in your mind, if you see that tree, where the ancient genome might fall. And surprisingly, we found it actually falls basal to 95% of the strains that are circulating in the world today that are human pathogenic. So the Black Death strain falls basically here on the root of this kind of little star, this kind of star-like explosion of different strains just here, very close to the most recent common ancestor. What does it tell us? It tells us the Black Death was probably the main event to actually spread bubonic plague in the world today, because 95% of all the strains that we have circulating in the world today go back to this strain. So in fact, this strain can be seen as some sort of mother of the plague. So it's some kind of like ancestral strain to most of the strains circulating in the world today. What we could also do is, because we knew exactly how old that strain is, because we know how old the site is, we know the site is 662 years old, this cemetery in London, we could use it to calibrate the molecular clock and tell us when did those different lineages diverge from each other. We could show it's the 13th century. So this is when the Black Death started, sometime in the 13th century, so a bit before it actually entered Europe, maybe somewhere in Eastern Asia. And we could also show that the entire human pathogenic as well as rodent pathogenic strains are only about 2,000 years old when they have their last recent common ancestor. So that basically the disease itself, bubonic plague, doesn't seem to be so old. So it has something that has probably evolved within the last few thousand years. So very much like the hypothesis that I presented at the beginning, that this is not a disease that is quite old. So in conclusions, what would we say? First of all, we could say Yersinia pestis was at least one of the causative agent of the Black Death. I don't say it is the causative agent because we cannot exclude that there might have been other pathogens spreading during that time. What we could also show, which was very exciting to us, was that in this ancient genome, we didn't find anything that was unique to the ancient genome that we do not find in the modern genomes. So it didn't have what we would call a private mutation or private substitution. So it has nothing special. It has nothing that we only find in the Black Death and that we don't find in the circulating strains that are existing in the world today. And that's an important piece of information because it tells us that basically all bubonic plague strains that are out in the world today could cause the same disease. So if we catapult them back into the medieval time, they would cause the Black Death potentially again. So that's an important piece of information. So it's still a very, very dangerous pathogen somehow. What we could also show is that it has evolved within the last 4,000 years when it has the last common ancestor with the outgroup, with Yersinia pseudotuberculosis. And like I mentioned, we couldn't find anything in the genetic makeup that could explain that it was somehow special and that it has caused so many people to die in the past. So basically it didn't have, at least based on the biology, a higher mortality, so a higher virulence than the strains that are circulating in the world today. So then, of course, there remains a lot of questions. And one of the questions that remains is, what then contributed to the high mortality that we observed during the 14th century? So why did so many people die? Why did 50 million people die in the 14th century, whereas in later outbreaks in the 19th century, for example, it was still 10,000s of people, but it was never again 10 millions of people. So people have speculated it could be climate change. So it could be that was the time when the small ice age started, so maybe the conditions have cooled down, which allowed pathogens to spread better. It could be changes in the vector population. So it could be that it wasn't the same rats or the same fleas transmitting the disease in the 14th century compared to the 19th century. And that we actually do know. We know that during the 14th century, the rodent that was transmitting the disease was the black rat. I don't know if you still have black rats in Scotland. We have almost no black rats anymore in Germany. They're actually on the red list. They're almost extinct. So the black rat is different from the normal rat that you might see close to the train station or somewhere else, which is the brown rat, which is kind of a rat, Rattus norvegicus, that only entered Europe in the 18th century. So it's actually a rather recent immigrant coming from Southeast Asia. So it was a different rat population, so it could be because the black rat was living usually within people's house. It was actually feeding like mice on grain that people were storing under the roof, that it was just living closer to humans, whereas other rats were living in the sewage, and maybe there was less risk of transmission to humans. It could also be that we have actually changed, so that if 50 million people die, if 50% of the population dies, that only the people survive that have certain immunity genes, so that have basically immunity genes that give them immunity against the disease, so that in future generations less people might die, so that mortality would go down. It could be that there were other disease spreading during the time, and then of course social conditions have changed. If you compare the medieval time to the 19th century, things have fortunately improved quite a lot. So I hope I could convince you that this is a very interesting field of research, ancient pathogen genomics. If I couldn't convince you tonight, I could at least convince some time ago the ERC, the European Research Council. They actually founded a project for us, which is called Ancient Pathogen Genomics of Re-emerging Infectious Disease, where we have now collected ancient skeletal remains from all over Europe that span about 2,500 years of human history. And we are now reconstructing pathogens from all those different sites. We have already published 2 papers. One was on leprosy, samples that were actually also from the UK, from Winchester. Some were from Sweden as well, and some from Denmark. And what we have also done in this project, we have not just looked at human pathogens. We have also looked at plant pathogens. We have recently reconstructed the pathogen that caused the Irish potato famine from ancient plant remains. That also worked pretty well and was also quite interesting to compare the modern pathogen Phytophthora infestans compared to the one that caused the Irish potato famine 150 years ago. One project that we have published recently, I still want to mention at the end of the talk, and that is a project which was very dear to me because it had a very, very unexpected outcome. And in this project, we looked at tuberculosis. So Mycobacterium tuberculosis, I think most of you are also familiar with this pathogen, is one of the deadliest pathogens we have on the planet. It kills about 2 million people every year, and 1/3 of the world population is infected with this bacterium. So even some of you here in the room might actually carry the bacteria. And most of you, it won't break out, but if you're immunocompromised, like you might have, for example, HIV or AIDS, then it could actually break out and might actually kill you. So it's a very, very severe pathogen. It has been hypothesized in the past that the pathogen is hundreds of thousands of years old, and it evolved with people in Africa. Other people have suggested in the past it was a pathogen that was transmitted from cows to humans because, as you might know, cows also have Mycobacterium tuberculosis, which is then actually not called Mycobacterium tuberculosis, but it's called Mycobacterium bovis, so the closest relative, the bovine form if you want. However, there was a problem when people looked where in the world you find different strains of tuberculosis, of modern tuberculosis in the world today. So you see there's a large diversity in Africa. There's those blue strains in Asia. There's the red strains in Europe. However, in the Americas, people only found European tuberculosis strains. And that doesn't make so much sense because America was settled by people coming from Asia. So it should have the blue strains, but instead it had the red strains. So what people then proposed was, okay, so Christopher Columbus brought it. So basically the Europeans brought the European tuberculosis with all the people coming to North and South America after the 15th century, and they were bringing tuberculosis, and this tuberculosis spread then all over the continent. So it's not a surprise that we only find the European form today. However, you have evidence of tuberculosis before Columbus being present there. You have lesions, you have bones that are more than 1,000 years old that show tuberculosis being present in the Americas. So, but that cannot be the European form. At least at the time there was no connection. That couldn't have traveled between Europe and the Americas. So that was a big question. So we wanted to find out, how did tuberculosis get to America in the first place, and what was there in the first place? What was the tuberculosis that was present in pre-Columbian times in the Americas? So there are different hypotheses. Like I mentioned before, it could have spread basically in younger times with Columbus, but then this is not compatible with the bone lesions that we have. So then it must be older, so it could have spread through Asia coming to the Americas. But then it should be an Asian strain. It should look like the ones that we have in Asia. So there was some problem here. It was not really compatible. So how could it have entered America from Europe? So we did the same approach that I showed you before. We used those microarrays. We used this kind of glass slides to capture DNA from ancient bones, actually from those ancient bones that you've seen here that show these typical lesions. We could get, again, millions of DNA fragments from ancient tuberculosis from those ancient Peruvian samples. And we were able, indeed, to reconstruct entire genomes again of ancient tuberculosis from those ancient skeletons. If you then look again at the family tree of tuberculosis here, you can see there are different strains, some that you find in Asia, some that you find in Europe, some that you find only in Africa. And there are also some strains that you only find in animals, because tuberculosis is not restricted only to humans, but you also find it not just in cows, but in many other animals. It was isolated from more than 2,000 animals by now, and it's probably in many, many more different mammals that you find this disease. However, what you can see in the family tree is that the animals and the animal strains, they branch off the human strains. And that was interesting. People have already observed that 10 years ago, that it seems like we humans actually transmitted the disease to all those animals. and not the other way around, like I hypothesized at the beginning, where I said from the domestication, we actually received pathogens from our domestic animals, but it seems like we have actually passed on the pathogen to all those animals. And now we have the ancient genomes from the Americas. We wanted to see how do they fit into this family tree, and you can already imagine where they might fall. They actually did fall together with the animals. They actually looked like animal forms. So that was a big surprise to us. What we could also do is we could then reconstruct the family tree more in detail, compare our ancient strains from Peru to the ones that we find in animals and the ones that we find in other humans, for example, in Africa. So first of all, we see it seems to have derived from those African strains. So it seems to have an African origin. What we, however, also saw was, what you see here is that the branch, this family branch tree here, the branch here is actually very short. It's much shorter than the other ones, and that is because it died 1,000 years ago. So it is missing time. It couldn't accumulate new differences, new mutations. And this is actually important information because we can use it again to date the common ancestor and date the emergence. So when did it actually evolve? So if we compare it to the root, to the most recent common ancestor, we have 1,000 differences between the Peruvian strains and the most recent common ancestor. Whereas with the modern strains, you have about 1,000 differences. We have 850 here, you have 1,000 there. And we know from radiocarbon dating exactly how old our ancient Peruvian strains are. They're actually 1,000 years old. So that tells us that per 1,000 years, we have 200 mutations. So if we have 200 mutations per 1,000 years, the most recent common ancestor of tuberculosis is then only about 5,000 years old. And that was a big surprise because most geneticists before said that tuberculosis is an ancient disease. It's more than 100,000 years old. It has evolved with people in Africa. It has left Africa with humans coming to Europe, to Asia, and later on to the Americas. So it seems the disease is only 5,000 years old. However, this creates a problem. If the disease emerged in Africa 5,000 years ago, how did it get 1,000 years ago to the Americas? There was just no connection. It couldn't have come over land because 5,000 years ago, the Bering Strait was already flooded. There was just no land connection. So we were really scratching our heads when we had this result because it really didn't make any sense to us. How did it get 1,000 years ago from Africa to the Americas? And how is it an animal strain and not a human strain? We really had a hard time to explain this. However, what we did, because we saw it was falling with animals, we sequenced more animals. So we looked at some more goats, we looked at more rodents, but we also looked at some sea mammals, like sea lions and seals, because we knew they can also get the disease. And we also looked at primates and apes, such as chimpanzee strains. So we got a whole bunch of more animal strains, so we did the phylogeny with those ones. And what we actually found, to our surprise, was that the closest relative of our ancient Peruvian strains were the ones that you find in seals today and sea lions. They're actually extremely close related. You would actually call them seal strains that we found in these people in Peru 1,000 years ago. And that is also found in the archaeology. If you look at those ancient pottery here, you see people having a lot of interaction with seals. They were hunting seals, they were eating seals. So seal was on the Peruvian coast one of the main protein sources. So they basically had a lot of interaction, and probably during those interactions, they caught the disease. And that then also explains how it could actually come over 1,000 years ago from Africa to the Americas, and that was probably simply by swimming, because we know that all seal and sea lion populations in the southern hemisphere, especially around the Arctics, have this mycobacterium pinnipedi, the one that causes tuberculosis in seals. So probably somehow over the Antarctic it has spread and then arrived in South America, and there on the coast infected humans, and those humans seems to have carried the disease into Central South America and also to North America a couple of hundred years later, spreading the disease. So it was actually a zoonosis from seals to humans, whereas again in the first place probably humans gave the disease to other animals in Africa. So it seems to be some jumping back and forth of tuberculosis between humans and animals. I want to mention again the new institute that we're building up, where we're trying to combine linguistics, archaeology, so ancient DNA, so archaeogenetics and archaeology into one new institute. where we're basically trying to reveal more about the history of humans, first of all, but also about the history of commensals, of domestic animals, domestic plants, so basically interactions, biocultural co-evolution over the last 10,000 years. Then I want to acknowledge my group in Tübingen, my PhD students, mostly Kirsty and also Alexander, that have done a lot of the work on the tuberculosis, and Yersinia pestis. A lot of collaboration partners from the States, also Julian Parkhill here from the Sanger Institute, and people in McMaster. Lots of other co-authors, funding agencies, and you for your attention. So thank you.
You were talking about the Black Death, and you said that the strains that we have now compared to the ancient strain, there's nothing really different. But how can you rule out that there aren't any novel plasmids in the ancient strain? Because you're using an array to detect them, right?
Sure. So it would be, of course, possible that there were plasmids in the past that has just been lost. However, we have a whole bunch of outgroups in this phylogeny. So we have the rodent pathogen. I haven't mentioned some other pathogens that are found throughout Asia. None of them have those plasmids. So basically also the more basal outgroups, they all don't have the plasmids. Then you have a large radiation. You have hundreds of strains that have been sequenced so far. None of them have another plasmid. So you would have to have a very funny thing in evolution. So we'd have to have a new plasmid popping up, radiating all over the world and disappearing all over the world. And that's usually not how evolution works. So this is very unlikely somehow that this could have happened. It's not impossible, I would say, but it's rather unlikely, I would say.
In the bubonic plague, do you know what the possibility of the reason why it sort of spread so fast was because of the virulence, whether the virulence was too high for humans, a bit like at the beginning of Ebola, if you know what I mean?
So
is that the reason why it's more virulent than...
So you're referring to the hypothesis that pathogens that are extremely virulent lose virulence basically over time. So which would basically, if a pathogen is too virulent, it kills too fast, it cannot spread, right? I guess that's the idea. However, it seems that the Black Death pathogen, this strain that we have analyzed, was pretty virulent, had a pretty high mortality, killed a lot of people, and still it spread all over the continent. And I think also with Ebola, people have argued for many years now that it can never cause an epidemic because it's killing too fast. However, we have just seen in the last couple of months a big epidemic. We're not even sure if we get it under control or not, even though it's extremely virulent. It kills within 3 days after the first symptoms actually appear. So it is actually extremely virulent, and it actually spreads amazingly fast. I mean, this funeral where this healer woman was buried, there was 3,000 people at the funeral, and 350 people got infected during this funeral because they just touched the dead body, and they basically just had like a few particles of the virus on them, and then they still spread it out. So I think it depends a lot on population size and basically density. So you have a lot of people living at the same place and in dense quarters. The pathogen still finds enough people to spread. And that's also probably the main reason why most of those pathogens have a rather recent history in humans, because human population has only really expanded massively in the last 10,000 years. When we had hunter-gatherers, people were living in small groups of 12, 15 people. Even if they picked up a pathogen from a bat or from a monkey or somewhere in the forest, maybe everybody died, but they didn't really have time to transmit it to the rest of the population. So pathogens had a really hard time in our past, but they find perfect conditions now where people live so close together.
Hi. I was just wondering about the Black Death in terms of, did you really just get one sort of sample site, considering that the Justinian plague might be also prior to that? Do you have no samples from other grave sites? And also about the tuberculosis, considering the seasonality of seals, surely that would be seen earlier in the New World? Or am I just misreading everything?
So it was earlier than...
later than 5,000, but earlier than 1,000 BP?
Yes. So for the first question, do we have different and other samples for the Black Death? And there's, of course, thousands and maybe 10,000s of skeletons all over Europe. in mass burials. And we are currently looking at a whole bunch of them. And there's many other labs that are looking at those ancient plague genomes also. And there's a number of genomes that are in submission. Some are published already. So there will be more diversity basically presented in the next couple of years. And there has also been some really exciting new insights because of those. One of the insights is that the pathogen that entered Europe, the Black Death one that I've shown, has actually moved back to China and caused the third pandemic in the 19th century. So that seems pretty apparent based on the genetic data that we have now. So it's kind of funny cycle. It comes from China to Europe and actually goes back and causes the 19th century pandemic. So that was quite interesting. So there will be more data, and it's just not so easy. I mean, you have to start somewhere, and that was the first start. And the good thing about the East Smithfield site is that it's a cemetery that was for sure associated to the Black Death based on historical records. And that's not so easy to find. London has fantastic historical records. Most villages somewhere in Europe don't, that go back to the 14th century. And just based on a burial, it's hard to see whether a person died of Yersinia pestis or just because that person had some other disease or was old. So that's not so easy to actually find the skeletons, or people just starved or something like that. But of course, we're looking at more material. The question about tuberculosis. So the seals have transmitted the disease probably earlier, probably sometime in the 5th or 6th century. At least this is where we see the earliest evidence of tuberculosis, which is like pretty strong evidence, at least, for tuberculosis in the New World. Surprisingly, we find the oldest evidence of tuberculosis in the New World in Southern America, not in Northern America, which is kind of consistent with the theory that it entered from the south and actually spread to the north with the seals, because in the Northern Hemisphere, seals don't have tuberculosis today, at least. So that's important. Piece of information. And we also dated the most recent common ancestor of our Peruvian strains and the strains that are found in seals today. And there we come to about 6th, 7th, 8th century. So that also makes sense. So that was the time when it basically spread from the other seal strains that you find today in Australia, New Zealand, Africa, as well as in the Americas.
I wanted to ask, you said that the bubonic plague came to East Europe by probably some dead bodies thrown over the
That's the
walls.
narrative, yes.
So is there any information if this was a problem earlier in China or what they did against it?
Yes, it's a very, very important point. It must have happened also somewhere in Asia, because this is where we find this huge genetic diversity in the bacteria today. And the ones that we had in Europe was really just one strain, or maybe two strains, but not as much diversity for sure as what we see today in China. So it seems there was a large pandemic also based on the modern genetic data somewhere in Asia, somewhere in East Asia probably, but we have no historical records. There's very, very few records that would suggest that there was something like the Black Death going on in China, even though China was denser populated at that time than Europe. It could be that the historical records from that time period are not good. I talked to some Chinese colleagues that were saying that during this dynasty, they didn't keep as good records as some previous and some later dynasties, but it's still hard to imagine if millions of people die that you wouldn't find that either in the archaeological, in the anthropological, or in the historical record. But there is no good account of that. And that's something we've just started a collaboration with Fudan University in China, where we exactly wanted to address that question, like what happened actually in East Asia during that time, because it must be there. I have a hard time to believe it wasn't there, but people haven't really found it yet.
You said the tuberculosis that you find in animals now seems to have come from humans, but if you've dated the human origin at 5000 BC, it sounds like humans might have got it from pastoralism, from farming, from the first domestication. So where would you look to find a tuberculosis in animals that could have been the origin for humans?
Yeah, it's a very, very good question. So why would it be 5,000 years in humans? Why has it evolved during that time? Now, one possibility, if you look at the effective population size of humans, it does two big increases. The one is basically in the last 500 years, where human population size kind of exceeds out of the roof. The other big leap in the human effective population size is during the early Neolithic. So around 5,000 to 6,000 years ago, the human population size increases. So when the human population size increases, like we just mentioned, that's perfect conditions for pathogens to spread. So that would be good for tuberculosis. Tuberculosis needs also larger population to actually spread within the population. So that could be the reason that it entered the human population during that time. Where we got it from, we have no idea currently. It seems pretty clear that if we look, just go back to the tree here. If we look at this tree, which is basically the kind of latest tree in tuberculosis, family tree, we see that what's called Mycobacterium canneti here, this is the outgroup. So this is basically the root of the tree, and then you have all those human branches branching off, and then from the branch 6, the animals branch off. So that's human, 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 and here come the animals, which tells us that we must have passed it on to animals, because at the root you don't have animals, at the root you have humans. So humans had it first, and they gave it to animals. So that's basically what comes out of the genomic data. And that's 10 years old. People have suggested that and shown that the first time, and we can just confirm that. So it's quite surprising. I still have a hard time to believe that. I still really have a hard time to believe that we passed it on to so diverse animals all over the planet, not just to our domestic ones, not just to goats, to pigs, to dogs, to horses, to cows, but also to seals and some monkeys and lions and buffaloes and dassies and all kind of creatures. So it's like, how is this working? Of course, it could be that we gave it to cows and they were eaten by a lion, and maybe they interacted like among each other, and like disease can just spread. I mean, maybe fruit bats or something else was spreading it. We don't really know. But we haven't found yet the strain that sits really on the root in some animal. Mycobacterium canneti itself can cause also tuberculosis. It's extremely rare. You only find it in East Africa. So the outgroup you find in East Africa, sometimes also in monkeys, but mostly in humans. So it could be that there was some reservoir, some host somewhere in Africa that we haven't identified yet. But it's a really good question, like what happened during that time, where did it emerge from, but currently we don't know. We haven't found basically the host, the wild host, where it could have emerged from. But it seems, at least from that data, pretty clear that we passed it on to all those animals that now have kind of the close related strains. They are not called Mycobacterium tuberculosis, they're then called Caprae or Bovis or Oryx or Pinnipedi or chimpanzee bacillus, but they are all on this one branch, the branches of a human branch. So.
Yep. For the Black Death, you talked about the bacterium. What about the host? Were you able to amplify the HLA, for example, and look
Yes.
at that side of it?
No, sure. It's a very, very important point also. Like what happened to us, and it's one of the kind of outlooks that I showed, like did we adapt? And that's a question that we're really addressing in our research currently as well. We have a large project, part of this ERC project, where we're looking at immunity genes, and not just the HLA, actually 250 genes, different gene clusters that are kind of part of our kind of inborn immune system. We are trying to look if we see changes in those immunity genes through time, before and after the plague, or in mass graves, do they look differently to the people that survived? But we haven't actually gone to the point where we really have results that are informative. There was a paper published which showed that at least one gene that has been for a long time hypothesized to be related to the plague, which is CCR5, which is a chymokine receptor 5, which is giving you resistance against or immunity against HIV. So you don't get HIV if you have a mutation in that gene. And the frequency of that mutation is about 10 to 15% in some parts of Europe. So it's really high in Europe. In the rest of the world, you almost don't find it. So it seems to have been selected in Europe. That is so common in Europe, this immunity gene against HIV. But the driving factor was certainly not HIV, because HIV only entered Europe 20, 30 years ago. So people then suggest it might have been the plague, so that this gave you not just immunity against HIV, but maybe also to the plague, which makes actually no sense if you think about the biochemistry of this specific receptor, but people have suggested that in the past. Other people have suggested this is wrong, has been a lot of debate. Now people have recently actually looked in the past, they have looked at medieval material, they find the same frequency in 11th century skeletons in Europe that we find in Europe today. So that hasn't really changed over time. So at least for this gene, we can exclude it. For HLA, it might be very interesting. It's certainly the most interesting part of our human genome where we could look at that. It's a very complicated locus though. So we have really spent and scratched our heads over the last 2 years to just get the arrays to represent all the genetic diversity you have in humans today on such an array is quite challenging, but we're working on it, and hopefully sometime in the future we have an answer for you.
Have you ever found yourself either critical of or supportive of the World Health Organization and the international health regulations, or are they too modern?
I mean, I don't interact too much with the WHO, and I mean, I don't really have a critical view on the WHO because, I mean, my epidemics are so kind of far back in the past that the WHO has no opinion about them. But yeah, so I can't really comment on that.